Before introducing Jan Henrik, I wish to give the floor to Claudia Vander for an important announcement related to the future activity of our Friday appointments. Claudia is the manager of the industrial board of Euro-BioImaging. It means the panel that gathered the companies interested to the Euro-BioImaging project. Claudia, the floor is yours.
Thank you, Silvio. So I just want to make a quick announcement and point out to you that we are planning to have a special edition of our virtual pub on April the 16th, where we are inviting flash presentations from both the academic community as well as from our industry partners in the industry board to tell us about your projects and your challenges when it comes to speed in biological and biomedical imaging. So what do we want to hear from you? We invite everybody to submit an abstract for short 5-minute flash presentations and just tell us your story about the fastest technology you've used in imaging, the fastest application you have done, or the fastest processes you have imaged in your samples and in your project. And to make this even more attractive, we will hand out a prize at the end for the best flash presentation. So please go ahead and submit your abstract until the 9th of March. And we are then looking forward to have a really entertaining and fun virtual pub in April. And I also put the link in the chat for everybody to click on.
Okay, thank you, Claudia, for the announcement. So I think that now is time to start our scientific session. And I am very pleased to introduce the speaker of the today virtual pub meeting, Professor Jan Henrik Ardenkjaer Larsen. Sorry, my pronunciation is not so, for this, if I did some mistake. Anyway, Jan Henrik is a full professor and head of the section of magnetic resonance of health technology of the Technical University of Denmark. He is also head of the Center for Hyperpolarization in Magnetic Resonance, a center of excellence funded by the Danish National Science Foundation. His research activity deals with methods to improve the sensitivity of magnetic resonance imaging and spectroscopy, with a dedicated focus on the dynamic nuclear polarization and its application to the clinical translation of hyperpolarization in medical imaging. Jan Henrik is internationally recognized as the scientist that brought the main contribution to make hyperpolarization by DNP the technology that we know now. So huge important in the field of imaging is the technology that allowed for the first time to detect in real time metabolic processes from a living system. For his contribution in 2012, Jan Henrik received the prestigious Gunther Lauken Award, and in 2019 he got the ERC Synergy Grant to do research in quantum hyperpolarization for ultrasensitive NMR spectroscopy and imaging. So I think that he is really the best person that we may have at our pub meeting today to introduce us about the huge capabilities of hyperpolarization in molecular imaging. Jan Henrik, the floor is yours.
Well, thank you very much, Silvio. Thanks for the very nice introduction and the invitation to present this work at this meeting. So I'll just give me 1 second. I'll see if I can share my screen. You see my screen now?
Yes, we see it very well. Yes.
Okay, excellent. And you hear me well?
Yes, very well. Yes, thank you.
Thank you. Well, again, thank you very much for inviting me to present this work in this seminar series. It's really a pleasure. And the title of the presentation is Hyperpolarized MR, modality in SPE. So this is really a new way of imaging, a new modality that is still at a research stage in early clinical development. So it is really a research field of its own at this point in time. In addition to what Silvio said in his introduction, I want to say that the idea, the invention behind this technique and the early research was done or started about 20 years ago, or even a bit more maybe, in industry. Back then, I was employed by Nycomed. That later merged with Amersham and finally was bought by GE Healthcare. So there's been a long industrial development and research in this field. And when we started this project many years ago, it was really with the aim of adding a new tool in medical imaging. It was the clinical possibilities, the clinical potential that really attracted us. And as you know, we need better tools, better diagnostic tools in order to understand disease. And metabolism, metabolic changes is really something that is underpinning almost any of the major diseases as we know them. We know that people are predisposed by their genomes to various degrees, but that this genetic predisposition is manifests itself as it's being, let's say, triggered or as a response of environmental factors and how we live, how we eat, how we our diet, our exercise, etc. All the factors that we are exposed to in our daily life. And we don't really know which person will develop the disease, which person where the disease will be triggered by these environmental factors. And we need better tools to understand these metabolic changes at an early stage in order to make a good diagnosis and give the patient the best possible treatment, a personalized individual treatment. So that was really the motivation for us when we started this project. And as you probably already guessed, let's say the physical principle, the modality behind hyperpolarized MR is magnetic resonance imaging. So, and this is one of the 4 major imaging modalities that we use currently clinically, at least to a large degree. You see them here compared in some way. If we look at the upper right, it's just to sort of put it into a context. Magnetic resonance imaging is widespread. It's widely available. We do a lot of procedures every year. Probably more than 100 million by today. It builds on the principle of detecting the magnetism of water protons. Typically, that's how we generate the image. So it's the magnetism of nuclei. Water protons are abundant in our body. It gives a magnetism which is sufficient to detect. And we detect radio waves in the 100 megahertz range. So radio waves. And then we code the space by magnetic field gradients, and that's how we generate the image. One of the other more important modalities is based on X-ray, so CT and transmission X-ray, and that's also very widely used. In the We will, in a minute, I'll go into what kind of images MR can do. So I'll not do a direct comparison to CT images, but CT is typically more first in line, easier to use. There's only one contrast mechanism, the attenuation of X-rays, whereas in MR we have many different contrast mechanisms, which is the advantage, but it's also more complicated and probably slightly more expensive procedure. And then we have the two others, ultrasound, which is easy to use, detects sound waves as they are reflected, propagate and reflects in tissue with tissue contrast, which is more portable, easy to use, more again also first in line, and also with let's say with practitioners in many different specialities, and where we do a lot of procedures. And then we know for detecting for molecular imaging, nuclear medicine is really a very strong player because of its very high sensitivity, the fact that we can detect trace amounts. We don't have any endogenous radioactivity, so we need to inject a contrast agent in PET and SPECT, obviously. In nuclear medicine, in ultrasound, we rarely use contrast agents, but in MR and CT, it's quite common to use contrast agents, and every third quarter to a quarter of procedures actually use a contrast agent to improve the contrast in the images. So these are the four modalities, and MR is really very suited for many situations. It is a modality that gives us images of very high quality, very high spatial resolution, very high temporal resolution. We get a lot of functional information, as you can see from these examples. The soft tissue contrast is excellent. So as you, for instance, see from these, let's say, brain CNS images, the soft tissue contrast is really excellent, and we can see many different pathologies with very good contrast. This contrast can be improved by conventional contrast agents to change the magnetic properties of the water protons that we are imaging. But this is extremely useful for diagnosing different pathologies. Now, this is all based on imaging the water protons, and that gives us a lot of morphology and anatomy. But as you know, magnetic resonance is used in also as a spectroscopic tool, because not only the proton has a magnetic moment, many other nuclei of the periodic table are magnetic, and we can in principle detect them by magnetic resonance. We use that in NMR for structural elucidation. And it would be really nice if we could also do this in vivo on patients, and we could, let's say, image or detect metabolites. This is, however, very, very challenging, primarily because we go from tens of molar of water protons to millimolar concentrations of metabolites. It's also partly challenging because we have so many different metabolites in our body that it's a very crowded spectrum. If you see this example to the left, it's a proton-based MR spectroscopy on the brain, and you can see from the parameters that we can actually detect and generate, let's say, single voxel spectroscopic spectra. Or images of the brain of the most abundant metabolites, and we can do that with reasonable spatial resolution and with reasonable acquisition times. So this is useful and used clinically in some situations, but it is complicated, and it's really hard to get the clinical penetration of MR spectroscopy. You also see an example here to the right where a patient has been infused with carbon-13 enriched glucose, a fairly high amount, which is then metabolized by the cells, and by sacrificing further spatial resolution and acquiring images over time, then we can see that the carbon-13 is incorporated into different metabolites as the glucose is metabolized in the cells. But since carbon-13 is even less sensitive than the proton, the sensitivity is further reduced, and we basically lose all the spatial resolution. So the idea behind hyperpolarization is that we are trying to overcome some of these limitations. Hyperpolarized means that we are creating let's say an artificial magnetization. We pre-activate our nuclear spins so that we have a strong signal. And in a bioprobe that could be enriched with carbon-13. This molecule enriched in carbon-13, it could be glucose as in the previous example, is then magnetized, hyperpolarized in this activation step, which I'll explain very briefly in a minute. And then we can inject our bioprobe into the living system all the way from cells to potentially humans. And then we can detect with magnetic resonance what happens to this to the molecule, and we see that this the magnetic nuclei then propagates into products of the metabolism. So this is what we are trying to. This is what we are researching. That we are trying to find suitable biomarkers that fulfill the criteria of hyperpolarized MR, and then translate them to clinical applications. That is the whole point. Now, just very briefly on this hyperpolarization, the challenge in magnetic resonance is that I've said a number of times is that we detect radio waves and in the 100 megahertz range. So the energies, the magnetic energies that we're trying to detect are weak. It's really nano electron volt, many orders of magnitude less than thermal energy. We're not it's not like nuclear medicine or optics that we can detect single photons. We need a lot of spins, a lot of molecules in order to have a decent magnetization that we can detect. And since the magnetic energy is so many orders of magnitude less than the thermal energy, it also means that these, the protons, for instance, all these nuclei are not really aligning very well with the magnetic field. So we have a state like this, typically that there's only almost a random distribution of these little small dipole magnets, the protons, or their magnetism, and only a very weak alignment with the magnetic field as we put them into the 3 Tesla or 1.5 Tesla MR scanner. We express that as parts per million. And what we would like to do is then to align all these nuclear spins with the magnetic field so that we get a strong magnetization, a much stronger signal. We do that in a polarizer. It's an activation step. In this polarizer, we take our, let's say, glucose, carbon-13 rich glucose as an example. We cool it down to a very low temperature, around 1 Kelvin. That reduces thermal energy. And then we dope into our sample an electron spin, which has a very strong magnetic moment. We can transfer polarization from these electron spins to the nuclei in the material, all the carbon-13s. And then when we get a very strong alignment of the nuclear spins in a solid state at low temperature, then we quickly dissolve the sample. So we get a liquid solution of the glucose. And in this phase transition, this temperature jump to room temperature, the nuclear spins stay aligned with the magnetic field, and we have a strong magnetization that we can detect in our MR scanner or NMR spectrometer. And you see here an example of urea, in this case, carbon-13 in urea, where we get a very strong signal in a single scan after the hyperpolarization, and where the same sample, after several days of averaging in our NMR spectrometer, only gives a very weak signal. So the hyperpolarized sample gives a signal enhancement of 24,000 times, which really allows us to do experiments that we couldn't do otherwise. It's a huge gain in sensitivity. And that's really the kind of enhancements of the signal that we need in order to go from molar concentrations of nuclei that we normally image on water to the millimolar concentrations of metabolites. So this is really the enabling step that allows us to follow the fate of the bioprobe as we inject it into the living system. So here's a short video of how this happens. So you see here a normal NMR magnet, superconducting NMR magnet, which can cool the sample in liquid helium. The sample has been sitting there for half an hour, 1 hour to be polarized. And now the two PhD students are ready to dissolve, to inject it into an animal. So they lift it out of the liquid helium, put a stick in and inject hot solvent. It dissolves quickly the, let's call it the glucose. And then they run to the MR scanner with their polarized substrate, and they inject it into the tail vein of the rat. And then it circulates by the bloodstream to various organs. In this case, they're studying metabolism of the heart. So there's a coil on top of the heart. You see the very strong hyperpolarized substrate, and then you see metabolites of this substrate appearing in real time. So metabolism in real time of the heart in a rodent. And this is the type of equipment that we developed early in the project. Just very briefly give you an overview of the different, let's say the development of the technology behind this. We did build the first prototypes many years ago, built a couple of clones of that. One went to the Oxford that you just saw in the video, Cambridge, a few other places. It also became a product by Oxford Instruments, which was discontinued 5 years ago, but these systems still exist in some places. GE continued the development for the clinical applications. So we took one to a clean room at UCSF, where it's used for the first patient studies, and then GE developed further instrumentation for clinical development. And at my lab at the Technical University, we developed the latest generation of polarizers that will eventually be useful for both preclinical and clinical hyperpolarization. And that is also now commercialized to enable research in different places. Now, as you guessed from the video, this activated state has a relatively short lifetime. So when we actually need the hyperpolarization to take place close to the actual experiment, the polarizer generates the hyperpolarized solution, which is then quickly carried to the MR scanner where it's injected, and then we need to image within a very short time because the magnetization decays quickly. So we inject by intravenous injection the substrate. It circulates with the bloodstream to the different organs. That happens quite quickly. We know that within half a minute to 1 minute, it's circulated the whole body. And then the substrate is extracted into the tissue and taken up by the cells and metabolized. And the metabolism, of course, has to be fast on the timescale of the decay of the nuclear magnetization, which is typically on the order of 1 minute. So we have this very small time window of 1 minute. So it's a demanding experiment in that sense, and has, of course, limitations also for that reason. There are many nuclei that are interesting. Most of the elements of the periodic table have a magnetic moment. Not all of them are interesting from a hyperpolarization point of view and from a biological point of view, but some are. And those that are such nuclei as carbon-13, nitrogen-15, possibly phosphorus. And then, of course, there are also some of these metal ions that could be incorporated into interesting agents for maybe more functional probes, bioprobes. But certainly for, let's say, biological molecules, then carbon-13 and 15 are the building blocks of most organic biological molecules, and therefore the target for much of our bioprobe development. Yes, and some of the agents that we have studied in initial are small metabolites, intermediates of energy metabolism, typically. I'll get back to some of the probes, but one of the first on this list is pyruvate, and that is really the first agent that has gone into clinical development. So that has actually been studied in humans. It could be enriched in the C1 position or the C2 position. I'll get back to that in a minute. It determines where the carbon label propagates in the metabolism. But it is a very central intermediate of glycolysis and energy metabolism. But also many other small organic molecules can be used as hyperpolarized substrates. For instance, fumarate, which will then be converted to malate, hydrate to malate. It could also be probes that are not metabolized, like bicarbonate, where we would study the pH in the equilibrium between CO2 and bicarbonate by quantifying those two metabolites. DHA reduction to ascorbic acid, acetate to acetylcarnitine, another keto acid, KIK to leucine, amino acids, and many carbohydrates can be used as substrates. So these are just examples of substrates that have been studied in vivo and where we've been able to detect metabolism from that substrate in vivo. Yes, and one of the big challenges, of course, for the clinical translation is to make this demanding technology acceptable in a clinical practice. So the polarizer that was developed by GE automated many of the steps that you saw the two students do, and also avoid handling liquid cryogens by making a closed cycle cryogenic system to get the low temperature, automating the sample preparation, and also ensuring sterility and quality control so that we know it's safe to inject into a patient. So these have been some of the challenges that we've had to overcome. Now, you spend the rest of the presentation on discussing some of the biological applications and one of the main substrates. And pyruvate has really been a very central molecule in the development of this technology because quite early in the research we discovered that this had very favorable properties for many reasons. From a biological point of view, it's very central in the glycolysis. It's the end product of glycolysis as glucose is broken down in the cells, and the entry point into the TCA cycle. So from the biochemistry textbooks, we know that pyruvate can be either reduced to lactate, it can be transaminated to alanine, or it can go into the Krebs cycle as acetyl-CoA. And if we label pyruvate with carbon in the C1 position, it would come off as CO2 when it goes into the Krebs cycle, and the acetyl moiety goes to acetyl-CoA. We could also label in the C2 position, and then the carbon label will go with the acetyl moiety to acetyl-CoA, and we could see intermediates further into the Krebs cycle. But C1 pyruvate was the first labeling position to be studied, and you can see here from the NMR spectrum that we actually have a signal that is unique for each of these molecular species, and that is really the key thing of NMR spectroscopy and magnetic resonance, that we can actually distinguish the chemical environment of the carbon label, and we can quantify these chemical species individually in our imaging or spectroscopy experiment, and thereby we can see the conversion and the flux of the biochemical pathway. And in the case of pyruvate, we're particularly interested in the reduction to lactate because it builds on the Warburg effect. So one of the first areas to focus on was in characterization of cancer, diagnosis of cancer, and response treatment, and so on. Here you see an early example of a tumor model in a rat. It has a tumor inoculated in the stomach. In this example, you see that after the tail vein injection of pyruvate, that the tumor takes up a lot of pyruvate. So there's the high uptake of pyruvate. Monocarboxyl transporters are upregulated in this tumor model. And we also see that there's a high conversion of pyruvate to lactate. So this is really a manifestation of elevated aerobic glycolysis, a hallmark of cancer, the so-called Warburg effect. And you see that also in the spectra from different regions. So this is the let's say the principle or the property that we built much of the early work on, and also the first patient studies. So this is the very first one done at UCSF with GE Healthcare, small patient population of 30 prostate cancer patients. You see here in the proton image the anatomy of the prostate. The coil has been placed in the rectum. And this is the prostate. You can see here from the proton image that there's a dark area in this T2-weighted image. So that's an indication of the location of the tumor. So morphologically, anatomically, we can see it in the proton image, but we cannot from this image say much about the actual cancer, whether it's aggressive or how it would respond to any kind of therapy. Now, this is just to give you a little idea of the timing and the type of metabolism that we saw in the prostate. We did just a spectroscopy in two different slabs, one through the normal prostate and one through the tumor region. You see here to the left that the bolus arrives into the prostate, but there's no metabolism in normal prostate, no conversion to lactate. Whereas in the slab that goes through the tumor, you see the bolus arrival of the pyruvate, but also a metabolism exchange into the lactate pool and a strong lactate signal that we can detect. This gave us information about the timing of the experiment. You can see it's very fast. This is intravenous injection into the arm vein of a man, and already 30 seconds after the injection, we see the arrival and the metabolism of pyruvate in the tumor, which is pretty amazing. So it just shows how fast both the biology, the circulation can be. So this was the information we needed to go into imaging mode. And here we see another case of these 31 patients. Again, a man with confirmed prostate cancer. We see it here anatomically on the proton image. And then at the optimal time point, let's say 30 seconds after injection, we can generate a map of the production of lactate in the prostate. And you see here that there are two hotspots in this patient, actually, which was surprising. So this firstly shows that the tumor generates a lot of lactate, but actually also that there was a node which was not a tumor that was not seen or known prior to this study, to this examination. And this patient, due to having the bilateral cancer, was actually upstage and went for radical prostatectomy, and this was confirmed by histology that there was bilateral cancer. So it actually even had an effect for this particular patient. So this was first proof of principle, and UCSF have continued to include more patients and study other patient groups. This is some of their work which shows correlation of the conversion of pyruvate to lactate with the grade of the tumor, which is really one of the very important clinical questions that all these men that have prostate cancer, since it's not a very aggressive cancer, they are on active surveillance, and they have come back for their annual checkup, and the doctor has to decide whether the cancer has developed and it has become a more aggressive form and there has to be some intervention. So it would be really important if we could have a tool that would give a more accurate prediction of whether the tumor is now becoming aggressive and intervention has to be made. Here we just see in this clinical data, that there is actually a strong correlation with the grade of the tumor, which is very encouraging from that perspective. And also they have looked at patients that went on different kinds of treatment, in this case on hormone deprivation treatment. And this patient had a very large tumor, had gone through previous treatments with not very much success, and then went on this hormone therapy treatment, and actually turned out to be a responder, and that was clearly seen in the loss of the lactate signal as the tumor, as the cancer was affected by the chemotherapy, which is obviously a very nice and important finding and tool to be able to detect responders from non-responders, and have the ability to change treatment for the patient if there's no effect of the treatment. This is published work from Cambridge, which also illustrates one of the important aspects, and really the I know from neither from Kevin's side nor from mine is this a question of let's say competing against very excellent other molecular imaging modalities like PET, FDG PET, which is extremely useful in many different situations. But there are also a lot of patients that where we do not have good enough results with PET and FDG PET. And here, this is a study in a mouse model where they compare in a CRC xenograft model with a receptor agonist, the response to treatment. And in this case, the nice finding was that the pyruvate to lactate was a very early indicator of response to treatment, much earlier than they saw response on the FDG PET. So in some cases, the hyperpolarized MR may have some additional benefits, both due to the specific pathways that we can look at with hyperpolarized MR, but also because of the intrinsic properties of magnetic resonance, that we can actually distinguish the products of the bioprobe that we inject. This is other work from the NIH, National Cancer Institute, Radiation Biology branch, also published a few years ago in 3 different pancreatic cancer models with quite different properties, patient-derived adenocarcinoma models in a mouse, where they actually also used EPR imaging to measure or map the oxygen, the oxygen concentration in the tumor tissue. And these tumors are known to have different degrees of hypoxia. And oxygen is extremely important in terms of choosing the type of treatment, whether chemotherapy or radiation treatment would be effective. So having these 3 tumor models that have different degrees of hypoxia was able to demonstrate this very nicely in this model system. Now, firstly, the first step was to, since EPR oximetry is not yet, or has some challenges in being translated clinically, this can be done in small animal models, but it would be very hard to do it clinically. But otherwise, it would be an extremely valuable imaging tool to have as well. But unfortunately, we cannot measure the PO2 in patients. So the first step was really to correlate this oxygen, this hypoxia difference in terms of the metabolism of pyruvate. And this is what you see on the upper right, that as we would probably expect, that depending on the hypoxia of the cells, that they would have a different metabolism, a different conversion of pyruvate into lactate, that would also be correlated very strongly with the hypoxic environment, and that the most hypoxic cells would also generate or convert mostly into lactate. So there's kind of an inverse relationship there, which is, I think, very intuitively understandable from the biochemistry of this particular of glycolysis and pyruvate. And from this data and these models, they could actually do studies where they treated these 3 different models with both a oxygen activated prodrug TH302 or radiation therapy. And as you can see from these plots, that if we take the most hypoxic, it has the best response to radiation therapy, whereas yes, it has the best response to radiation therapy, whereas the most oxygenated tumor has a very good response to this oxygen activated prodrug TH302. So it responded very well to the chemotherapy due to the presence of oxygen, which we could have detected by EP oximetry or by the hyperpolarized MR experiment. Another area would be in imaging brain pathologies. This is a patient brain tumor image, which just demonstrates that we can also detect the metabolism in the brain. The brain is quite particular because we have the blood-brain barrier, so it puts a lot of additional constraints on the let's say, time constraints on the imaging experiment. But it's something that I personally find very encouraging, not only for studying brain tumors, but also studying other brain pathologies, neurodegenerative disease, and looking at changes in metabolism in the human brain. But I show this mainly to show you that this is clinically translatable, not only in the case of the prostate, but also to the head and brain and other organs, as we will see in a minute. The last example I will show you is for cardiac imaging, because also cardiac disease is extremely important from a patient point of view and associated with metabolic changes. So this is a study we did many years ago in a pig model, an occlusion model to create an ischemia reperfusion model. So a balloon was placed in the left circumflex to occlude the artery and create the ischemic region, and then reperfuse after. And what we observed and was that pre-occlusion, we can generate nice images of the myocardium. Now I just show you the metabolites. In this case, it's actually not lactate so much that was indicative, but more the alanine and the bicarbonate, so the flux into the Krebs cycle, because the heart is very energy demanding and actually metabolizes much of the pyruvate through the Krebs cycle. But by creating a mild occlusion, you can see that there is a defect in the bicarbonate image, a very small defect here, whereas a longer occlusion would give a much stronger defect in the metabolic image. The interesting thing is that if you compare with a conventional MR, if you look at the normal proton image or the perfusion image after gadolinium injection, delayed enhancement, then the short occlusion is reversible and does not create any damage to the myocardium. But still, we can detect this, let's say, decrease in metabolism in the ischemic region. For a longer occlusion, of course, we see it easily with MR because now we have scarring and we have a perfusion defect and, of course, very strong perturbation of the metabolism. But the interesting part is, of course, that we can detect the early and mild occlusion, mild ischemia, where the damage is still reversible. And finally, just showing that also is very clinically translatable. This is work from Sonny Brook, Jack Cunningham's group, where you see the cardiac images in a normal patient, and you just see that the image quality is really good. Pyruvate in the heart chambers, the nice bicarbonate image of the myocardium, and a lactate image, which is more diffuse because lactate is quickly transported into the bloodstream. And you see very nice spectra of the heart, where we can quantify the different metabolites. So it's really this kind of spectroscopic information that allows us to map the individual metabolites. This is also a very important area of research and clinical interest, and several groups are interested in studying cardiac metabolism in, for instance, patients with diabetes and other disease of the heart. Now, I talked a lot about metabolism, but personally, I have one of my focus areas is also on agents that are not metabolized, just using them as contrast agents. And in this example, you see actually that we can also hyperpolarize water protons and use that as a contrast agent. It has two aims. One is for angiographic imaging, because the hyperpolarized signal is so strong that we get really very nice images under the right conditions, image quality that is comparable to CT angiography, which is the way to image the coronary arteries of the heart. We want to see occlusions at the more distal part of the vessel structure. And the nice thing about this is also that we can actually look at the perfusion of the myocardium. So in addition to angiography, we would also be able to quantify perfusion of the myocardium, which in a similar way as we for instance, could do it with oxygen 15 PET, so it's really nice to get both of these two things in the same examination, both the angiographic visualization, but also and also the perfusion of the myocardium. Okay, and with that, I hope I have convinced you that hyperpolarized MR, hyperpolarized metabolic MR, provides a potential for staging disease and monitoring treatment response, characterizing disease clinically in many relevant diseases. Metabolism is really fundamental and underpinning many of the diseases that we are struggling with in society. The good thing about MR is that it doesn't expose the patient to any ionizing radiation, so we can actually examine the same patient at multiple time points and have follow-ups, and also in patient populations that where we would not use ionizing radiation, and that we, due to the inherent properties of magnetic resonance, can get detailed information about the biochemical pathways. We have been able to translate this to the clinic, and the first studies have been performed, as I pointed out, but there are several other papers that are out now from clinical studies in different patient populations. So now, of course, the big challenge is to do the formal clinical studies and find the right indications and introduce this as a widely in the clinic, which is not insignificant. But that is where the modality is. So thank you very much for your attention, and I'm happy to answer any questions if there are any. Thank you.
Thank you, Jan-Erik, for this very, excellent survey of the field. That show such an important achievement, and even more important, it show the huge potential of this approach. Thank you for showing how the hyperpolarization can be, can compete with other existing imaging modalities, but meanwhile it adds, I would say, something that other imaging modality cannot do. And this is particularly true in terms of imaging of the metabolism, by the way. I still think that in future, we would have, through this technique, the possibility to know a lot about it. So I'm asking Marianna to follow if there is any question. Maybe meanwhile I can start. You show, of course, the terrific importance of hyperpolarized pyruvate, because it is at the gross link of several metabolic pathway, and it has a peculiar characteristic that are shown to be, let's say, the metabolic biomarker of reference. But I wonder, in your view, in the field, which could be the other metabolic biomarker that you envisage in the near future that will accompany the huge success of pyruvate?
Yes. Yeah, I mean, if we look at it from a clinical point of view, then, well, so, as you obviously know, there are many constraints. So to take a probe into the clinic is quite a demanding task. But I think actually, if we just look at the technical limitations, the time window that we have available, the logistics, and so on, I still think that there are a number of agents that are clinically relevant. So in addition to pyruvate, fumarate is one that is on the list, the fumarate to malate, which I didn't talk so much about, but which is a very nice indicator of cell death, of necrosis, as shown by Kevin Brindle and his group. So I think that could be a very important marker. There's been attention on bicarbonate, because if you could image pH, that's also really a very has been a target in the research field, not only hyperpolarization, but in the general research field for many years. It's a very important physiological parameter if we could assess that clinically. There are some, urea has been proposed as a perfusion agent. It would not be metabolized, but is very well tolerated, and it could be a very good perfusion agent. And then possibly agents like KIC, ketoisocaproic acid, which also targets a very important transamination step in the metabolism. I think that could be very key, for instance, in the brain and in neurodegenerative disease. Glucose is something that we've studied, and of course, glucose, like with PET FDG, glucose would be extremely important. The question is whether it reports on anything different than the pyruvate, but for sure, glucose uptake and some of the intermediates of glycolysis, which we can see. Certainly actually it's been demonstrated in cells and in small animals, in mice, that we can actually see all the 10 steps of glycolysis if we start from glucose. So this needs to be understood better and studied, and could therefore also be a potential marker.
Thank you. I agree, fully agree. Are any questions from the audience?
I have a question. Thanks, Jan. Excellent presentation and broad spectrum of extremely impressive technologies and findings. I had two small questions. So first of all, regarding the contrast agents that have been used in the old days, when it comes to MRI contrast agents, there were some problems in terms of toxicity, or at least health concerns.
Yes.
So how is the state nowadays? And then I think it may have been in some of your slides, but I was interested in what kind of resolution can you, I mean, of course, on an organ scale, the resolution looks very, very impressive. But going down on really tissue level and maybe even cellular level, what kind of resolution can you achieve in the best of cases?
Right. Yes. Thank you. Yeah, I mean, for your first question regarding contrast agents, I mean, gadolinium-based contrast agents are still used clinically to a very large extent. Certainly, the toxicity associated with gadolinium that was discovered a few years ago has reduced the use of gadolinium, but it's still used where it's, where it's indicated. It's very important to get the best possible contrast. Certainly, the clinicians are more conservative in their use, and much of the let's say issues in the past has been associated with double and triple dose injections. Now there's a contraindication for patients that have poor renal function, so an EGFR criteria, and that seems to have solved the problem. But certainly there's a huge interest in finding alternatives to gadolinium, which is actually the title of our Fed Open project, where I do the hyperpolarized water, that we're looking into other alternatives, alternative agents. So that's just a little bit of a general information to your question. Regarding the spatial resolution of this particular modality of hyperpolarized MR, then even with the hyperpolarization and the spatial resolution that we can achieve in these metabolic maps would probably be very similar to what you know from nuclear medicine. So like if we take it clinically, for instance, we expect something like 5 millimeter isotropic resolution in a patient. In a preclinical, it could be done better, but it's not high resolution images in the sense as we know them from proton MR images. We have that independently. We have the anatomy, morphology from the proton image, and on top of that, we can then overlay the metabolic maps. That gives us the navigation, the localization, but the actual resolution of the metabolic maps will not be better than a few millimeters to 5 millimeters clinically.
Thanks.
Are there more questions from the audience?
Yes, Jan-Hendrik, just to thank you for a very nice talk.
Thanks.
My name is Ardenkjaer Larsen, by the way. I was just wondering, a follow-up about the spatial resolution. Do you see any prospect to improve that? Because as I see for the prostate, it's not much better than a normal proton spectroscopic imaging, and even less than the fusion-weighted imaging, in which the values, ADC and metabolic ratios, are giving you the same information as you now get out of this KPL. And the KPL is also kind of subject to a lot of factors. So what is the ultimate goal that you see there for in these cancers like prostate cancer?
Well,
if other modalities, as I see it, can give similar results and which are much more practical.
Yeah, no, that's really, thanks, Aaron. That's a really good question, and I mean, you're probably more qualified to answer that than I am. Actually,
Yeah.
I know from my naive, I'm not a clinician and certainly not specific on prostate, but the way I think of it is that it's really a question of specificity, sensitivity, specificity in the end. And I think that the DBI fails in MR spectroscopy, fails in too many situations clinically. And therefore we need something which is more accurate. And the hope is, and I don't know exactly whether, and that's also the reason that's this, let's say, staging or aggressiveness staging that I was talking about, and that we originally thought of as a good target, has been maybe a little revisited, and maybe it needs to go into more subgroups of prostate cancer where it makes more sense. But I really think that it's a question of having demonstrating that we are much more accurate than DBI or MR spectroscopy, which hasn't seemed to make the cut.
Yeah. That's very difficult, but of course. So, but do you see any prospects in improving the signal to noise so you can go to smaller volumes?
Yeah, I mean, well, we from a technical point of view, we definitely, I see several ways of improving the image quality. And we're certainly working on improving the polarization, where we make good progress, and I think we are superior to what has been done in the past. You also know that the coil development is extremely important in magnetic resonance imaging, and there's, I mean, decades of work has gone into optimizing proton imaging, but getting good X nuclei or carbon coils is very demanding and not easy to get hold of. So there's a lot of potential there. And then also in terms of doing this rapid spectroscopic imaging, there's been development in different types of different acquisition strategies where you can using compressed sensing, prior knowledge, model-based fitting, super resolution. So there are many of these known concepts that can be invoked, and where I think we can get small gains. So I'm very encouraged that we are actually able to get what we have seen so far, and from already at this stage, and there is an upside. But I don't want to oversell it either. And I think that we are now at a point where we have to show and demonstrate that we actually have a method that is more, I mean, is superior or equivalent to gold standards in some, or fills a gap where there is no in some clinical indication. Otherwise, it will stay a research tool, which is fine, but to have the clinical impact, it's
yes, sure. Yeah, I think there's also a future in doing artificial intelligence applications. So I was quite surprised that by room temperature carbon-13, you could, by denoising, you could still go to pretty small spatial resolutions and time resolutions, but
right.
Seem to be competing with DNP, but I don't think so. At the end, the DNP will turn out to be more sensitive. Okay, thank you very much for your answer.
Yes, thanks, Andy.
Well, thank you very much, Jan-Erik, again for the excellent presentation.
My pleasure.
The question and the discussion. Very useful to show how, his capabilities that this modality will contribute to the overall field of imaging of biological systems. And we were very keen, to see that soon, also in Euro-BioImaging, we will have that possibility to offer to our users this the state of the art of this imaging modality. Thank you again. And for closing this section, I will pass the floor to Marianna, who is our communication officer, that she will present then the calendar for the next events of these virtual pub meetings on Friday afternoon.
Thank you, Silvio, and thank you, everyone. I just wanted to invite you to our following pub events. So next week, we're going to be hearing from Dr. Ricardo Henriques of Portugal about open cutting-edge super resolution and machine learning technology for the study of cell biology and viral infection. You can see the lineup for the next coming weeks as well on this slide, and we hope to welcome you again soon. Thank you for joining us today.